Termination of neurotransmitters is pretty important for the whole process of excitation of downstream cells. When I say downstream cells, downstream of the neuron that's releasing the neurotransmitter. So that downstream cell could be another neuron, it could be a heart cell, a skeletal muscle cell, a host of other cells within the body. So I'm going to take a look at this. Acetylcholine is the example we're going to use for this. Here's a neuron releasing its signaling molecule or neurotransmitter known as acetylcholine via calcium-induced exocytosis. The acetylcholine is going to bind to receptors on the downstream cell. So once again, this is whatever cell you want it to be. Acetylcholine binds to nicotinic receptors in this example, which is a ligand-gated channel. We previously talked about muscarinic receptors that are G protein link receptors, but in this example, this would be representative of a nicotinic receptor, opens a gate, sodium moves down its electrochemical gradient into that cell to cause a depolarization event, which will result in further activity in that downstream cell. The gate does not need to be open very long to allow a sufficient amount of sodium into the cell. That is to say, enough sodium to come in to depolarize that cell up to positive 30 millivolts. That gate really is only open for a very limited amount of time, partially because acetylcholine immediately pops off of that binding site due to some enzymatic activity. There are enzymes known as acetylcholinesterase, oftentimes referred to merely as cholinesterase, that breaks down acetylcholine. When it breaks down acetylcholine, it's no longer bound to the receptor, the gates close, and that downstream cell is fully depolarized. But what happens to the acetylcholine that is broken down? Keep in mind, acetylcholine is a combination of two molecules, one known as choline, the other one known as acetyl coenzyme A. Acetyl coenzyme A is an intermediate of the citric acid cycle, which we'll talk about in a subsequent video, but that's the pathway of cellular respiration, or I should say the intermediate phase of cellular respiration. But the choline can be recycled back into that neuron that released it, in this case, this green one. So that choline can be used again in this neuron to make some more reconstitute acetylcholine. And within the cell, there's going to be mitochondria that are producing ATP, that are undergoing the Krebs cycle, producing more acetyl coenzyme A. So acetyl coenzyme A is going to continually be made within the cell. Then all we need to make more acetylcholine is the choline, half of that, and that is recycled from the synaptic cleft right here back into the neuron that released it. So if we look at this in red here, this is the choline. We see in black here, this is acetyl coenzyme A, which is going to be taken care of in another manner. But the choline is going to be recycled or be reuptaken into this neuron. So acetylcholinesterase breaks down acetylcholine, and then a sodium cotransporter is going to pump choline back into this neuron against its gradient. That's why we need the kinetic energy of sodium in this cotransporter to move choline back into this neuron. That is how acetylcholine is terminated, and half of it, at least, is recycled into the very neuron that released it. Norepinephrine is similar, except norepinephrine is reuptaken back into the neuron that released it entirely as a whole. That is to say, norepinephrine is just reuptaken by secondary active transport similar to the choline via a sodium cotransporter. The difference here is that acetylcholine is broken down into choline and acetyl coenzyme A. Norepinephrine is not broken down. It's just pumped directly back into the neuron that released it. Okay, that's termination of neurotransmitters.